Well, let me start my talk by saying happy birthday, amazing Bengt. Last night we were treated to his charming wife's singing a song about him, and I think it filled all of our hearts with joy because we all feel the same way about him. So happy birthday. So you've been hearing about DNA, which is a long linear code of life. It's a long linear template. And what it does is code for proteins. And when proteins are first decoded from the DNA, they're also long linear strings of amino acids. And they have to fold into these very, very, very particular shapes in order to do what they're supposed to do. And basically proteins are us. Just about everything that makes us human beings is due to proteins. In fact, all life on Earth. And the difficulty is that these long linear strings have to fold just exactly right in a crazy environment. They have to do that inside of a living cell where the concentration of other proteins is 300 milligrams per mil, and where there's a tremendous amount of kinetic energy. So this is a wonderful animation by Adrian Elcock that's based upon real protein structures and the real crowding inside of a cell. There's two ways in which, however, this movie is not realistic. One of them is that it doesn't convey, it has each of those proteins acting as though it's a solid thing. Each of those things that's moving around. And actually, in reality, they actually breathe quite a bit. They change conformations. They interact with each other. They do all sorts of stuff. And the other way in which this isn't realistic is that to be at the realistic speed at which this is occurring inside the cell, you'd have to speed that movie up 3 million-fold. So it's actually just plain inconceivable to imagine how fast proteins are moving around the cell and doing things. And this really is part of life. It informs all of life. All biological systems have evolved in this way, and it makes life possible. But it also creates a lot of problems in terms of protein folding, and especially with respect to how protein folding is influenced by changes in the internal and external environment. So you can imagine, for example, at slightly higher temperatures, everything speeds up a lot, but it's an exponential effect in that crowded environment. And there's all sorts of other things that influence protein folding, osmotic stress, the energy balance of the cell. So this really this affects every single aspect of biology you can imagine. And I've really been fortunate enough to start out at an early stage in this field in terms of the protein folding in the biological systems, and have been working on all kinds of things like cancer and neurodegenerative diseases, which are caused by misfolding, and the evolution of drug resistance, which is strongly influenced by protein misfolding. What I'm going to tell you about today is one way in which this protein folding problem winds up influencing the very nature of the way organisms evolve on this earth, the way they acquire new traits. And at the last Molecular Frontiers Symposium, I actually talked about one way in which that happens, and that involves a protein chaperone, a protein folding agent called HSP90, which binds and helps to fold a variety of different signal transducers, such as the androgen receptor, in fact, but many, many other signal transducers. And because of it interacts with the protein, it's a protein folding agent, and the way in which it interacts with the environment has profound effects on the way in which genetic information is decoded into biological traits. But I'm going to tell you about something different today, a completely different way in which protein folding influences the evolution of new traits. And in both cases, the message is that it provides a mechanism for the environment to create new traits. And so just want to frame the talk by mentioning this extraordinary man, Dobzhansky, who really transformed our understanding of evolution, really putting a molecular framework on the Darwinian theory of evolution back in the 1930s. And everyone in biology knows this quote. We all use it all the time. It informs everything we do. But he also said this: when it came to the inheritance of environmentally acquired traits, this question has been discussed almost ad nauseum in the old literature, so that we may refrain from the discussion of it altogether. And as I'm sure you students know, Jean-Baptiste Lamarck has been kind of held up for ridicule and all of the biological textbooks. And what I would like to do is convince you that in addition to the work I presented last time on HSP 90, this work on prions, it's really true that the environment can cause the inheritance of new traits, and it's time to give him back some of his dignity. So, my peculiar story is going to start with a very bizarre neurological disease. Disease known as Kuru in the cannibals of New Guinea, Creutzfeldt-Jakob disease in human beings, mad cow disease in cattle. And this disease was first shown to be a transmissible neurological disease by Carlton Gajduszczyk. And then it was Stan Prusiner who really trumpeted the concept that maybe it was a very peculiar properties for the infectious agent, just plain peculiar. And he suggested it might actually be the infectious agent rather than having DNA or RNA in it, might have been due to a protein with self-perpetuating properties that changed conformations and perpetuated that state, spread through the brain. And so prions have gotten kind of a bad rap. It's a really exciting concept in biology, but they're thought of by just about everyone as being really just negative, awful disease things. And so what I'd like to suggest is that you should think of this thing as a prion and say it's time to get rid of some of the baggage. Because prions can actually be amazing. They can create an extraordinary diversity of new traits. We've studied them, of course, the most intensely in a simple microorganism called yeast. But we know now that they're creating all kinds of interesting biological properties in higher organisms too, and we're just touching the surface of that. So some great things about prions: they provide a mechanism for proteins to act as genetic elements of inheritance. We've found 50 of them in yeast. There's actually 25 that we've published on, and several other people have published on about 8 or 9 of those. They create dozens and dozens of new traits. They help promote survival in fluctuating environments where being able to access different traits can be very, very, very helpful. They provide a very fast route to the acquisition of very complicated traits. One of the conundrums of evolution has been, how can we evolve complexity? If in order for each individual genetic change to accumulate and create a new trait, it's kind of hard for that to happen. Prions are just one of the ways, as HSP 90 is another, in which organisms can quickly acquire complicated new traits. So that creates a very sophisticated bet hedging mechanism in terms of being able to survive in complex environments. These prions are a mechanism for biological memory, and in fact, it turns out some work we did with Eric Kandel, we helped Eric Kandel with, but was really primarily Eric's and Cow6C's. This mechanism is actually a really cool mechanism used to self-perpetuate memories at the ends of synapses and neurons. It allows for life in complex communities, and that's going to be the final, ultimate Lamarckian piece, because there are ways in which bacteria can induce prion elements in yeast that cause both the bacteria and the yeast to live in a much happier way in complex environments. So here's the prion. The cells on here and the cells over here are genetically absolutely identical. They obviously look quite different. But when you streak out take up this red colony and were to restreak it, that'd be very, very, very stable phenotype. Except you see that once in a while some of the cells will turn white. And over here, it's harder to see the little red ones coming up on the white plate, but the same thing happens. It's kind of a metastable trait. It changes about 1 in every million, 1 in every 100,000 cells. And it has a very odd genetic behavior. And I want to give tremendous credit to Brian Cox, who started to figure this out and was doing experiments on this thing that everybody thought was really weird and he should go away and work on something else. But what Brian found was, as just one example of the odd genetic behavior, if this trait was due to a change in the DNA, then when you mated a red cell and a white cell, and then you got progeny out of that, you'd expect half of them to get the red chromosome and half of them to get the white chromosome, and so it would segregate 50-50. That's not what happens. You made a white cell to a red cell, and all of the cells become white. All of the progeny become white. Prion also was the one to figure out that it had something to do with a change in translation termination. So translation is the process by which messenger RNAs are decoded into proteins, really, really central and important process in biology. And Prion realized that that change in red-white color was because of a change in translation termination, and I'll explain that in a little bit more detail in a moment. But this protein, which we now know as a prion protein, has a peculiar structure to it. It's got this region here, which acts as the translation termination activity. And what I mean by that is it's the protein that tells ribosomes to stop when they hit a stop codon. And it's attached to this really weird thing. It's got a very peculiar amino acid sequence, a very, very highly charged domain, and a very glutamine and asparagine-rich domain. And this thing controls all of its odd genetic behavior. And this was discovered by a large group of people working on it. And just to give you one example of some of the peculiarities of it, if you get rid of this domain, the translation termination factor still functions just fine, but the cells can never turn white, and it can never access the difference in translation information that would make them white. And moreover, if you take this protein domain here, this weird one, that's responsible for, if you just simply transiently overexpress that protein, the cells will turn from red to white, and then you stop expressing it, and they will stay white, heritably change. So transient change in a protein causes a heritable change in the biology. So Reed Wickner first suggested that this might be due to this difference in trait might be due to some kind of a self-perpetuating difference in the property of this protein. And that really was hit home to Yuri Chernoff and I because we'd already been starting to work on it for the following reason. Yuri was working in Sue Liebman's laboratory, and he'd been looking for things that affected the inheritance of that switch from red to white and the inheritance of that white state. And what he found, the only thing in the genome he found that really mattered was a protein called HSP104. I was working on HSP104, so he called me up and said, do you know what this thing is doing? How is it controlling inheritance? And I did know what it's doing. In fact, one of my postdocs and I knew what it was doing. And we were the only people who knew what it's doing because we couldn't get our paper published on this for a very long time. So this paper was sitting on my manuscript sitting on my desk, undergoing yet another revision. And I'll tell you about HSP104, and it provides a very, very interesting explanation for the inheritance of that trait. So this is something called thermal tolerance, something I'd worked on for a long time. And basically what happens here is that you take two portions of a yeast culture, grown at 25 degrees, and one of them you put for a short period of time at 39 degrees, and then you put both of them at 50. And these are the guys that got that pre-treatment at 39 degrees. Tremendous change in survival. And after a lot of work, my lab showed that this tolerance factor turned out to be HSP104. And moreover, what we hadn't been able to get published was the way HSP104 rescues the cells is by disaggregating proteins that aggregate in response to heat. So you all know about proteins that aggregate in response to heat. This is one of them. But it turns out that just a little bit of that causes disaster in biological systems. And what HSP104 does is not to unfry the egg, but as little aggregates are accumulating, it can actually take them apart. And people just didn't believe that proteins would be able to take apart aggregated proteins, but it does. So I was very fortunate that my paper had been rejected because I got called by Yuri at a very interesting time in terms of trying to figure out how this inheritance worked. So the fact that HSP104 we knew was involved in controlling the protein's foldedness of certain kinds of misfolded proteins caused us, of course, to wonder whether the difference in the inheritance of that trait might be related to a protein aggregate. And so we labeled that protein that other people had identified, the translation termination factor, and sure enough, in the red cells, it's soluble and moves throughout the cell and does its activity on the ribosome, stopping ribosomes when they're supposed to. But in the white cells, most of it is actually tied up in these aggregates. And so ribosomes start reading through stop codons, and it changes the translational activity of the cell. But it's not just any old kind of aggregate, by the way. It's an amyloid. And that gives it some very peculiar properties. So that central domain of this amyloid fiber, these are protein fibers. The central domain is that weird amino acid composition protein, the portion of the protein that's responsible for the inheritance. Outside of it is that translation termination activity. Once it goes into these kinds of amyloid states, that translation termination activity can't get to the ribosome. And one of the key elements of how this thing works is the following. If you isolate that protein in a soluble state and sit it in a test tube, it will slowly assemble. And it assembles in a kind of peculiar way. Nothing happens for a long time, then finally it really takes off. One key to this whole thing is that if you take a small amount of this protein and add it back to the start of a new reaction, that's what's happened. So if the protein is in the soluble state, it tends to stay in that soluble state, cell division after cell division after cell division. Once it gets into that, once that amyloid has been templated, it can very quickly pull other protein in the cell out into that amyloid state that sequesters that protein from the ribosome, changes the way the ribosome is translating the protein. Cells switch from red to white. And the cells are inheriting then a new trait, not because they've inherited a change in any nucleic acid, but because they've inherited a protein aggregate that templates itself and it perpetuates that fold. Now, so how does HSP104 fit into all of this? Well, HSP104, as I told you, disaggregates proteins. And it can actually cut these amyloid fibers. And this is an example of HSP104's activity cutting amyloid fibers into small little bits that can be conveniently inherited by the daughter cells. So this is actually how this is working. This is a cytoplasmically inherited trait. That's why it's passed down to all the progeny. It doesn't follow the chromosomes at all. It follows, once this cytoplasm has mixed with this cytoplasm, it templates that protein into the new state, and it self-perpetuates its inheritance. Now, it'll be lost every once in a while whenever this template just happens to be not transmitted very effectively into the daughter cell. So putting it into a little bit more molecular detail, here we have the ribosomes reading along a messenger RNA and stopping at the stop codon of this protein that switches cells from red to white. And this translation termination factor is interacting at that stop codon and telling the ribosome stop. That's normal. But when the protein assembles into a prion amyloid, there's enough to stop many ribosomes, but not to stop all of them. So the ribosomes start to then move through into this downstream sequence. They make a new protein, a different protein, and that turns the cells from red to white. And then the second major piece of this puzzle in terms of inheritance is that HSP104 comes along. It cuts those fibers so that they can be inherited by the daughter cells. And it acts in effect as a sophisticated mitotic apparatus, just like microtubules segregate chromosomes. HSP104 segregates this protein-based information template that will create a new trait in the daughter cells. Now, is this just some weird oddity of red-white yeast? Why has this been preserved? It turns out this has been preserved, this domain has been conserved both for its ability to misfold into this amyloid state and for its ability to be cut by HSP104 for 800 million years, which would suggest that that ability to get into that amyloid state and to inherit that altered activity of the translation termination factor must serve some useful purpose. Why would cells want to have translation termination activity altered? Well, if you think about it, ribosomes start to move into that new information downstream of stop codons. They're not going to be doing it just on this red-white protein. They will be doing that on lots of other messenger RNAs too. So that then when this prion forms, it gives cells a chance to sample what's downstream of stop codons. And when it does that, the information that's downstream of stop codons is different in each strain because it hasn't been under selective pressures. It's been free to mutate because it doesn't normally matter. But once the cells switch into the prion state, now that information can matter. So that was our hypothesis. So we asked, okay, let's fix the red-white business and make the cells all white no matter, we'll fix that protein. But let's ask, do we see new biological traits if instead of growing the yeast cells under normal, great, happy media so that they're growing like crazy, let's stress them a little bit and put them under difficult conditions. So for example, and we set up lots and lots of different strains, but here's an example of a strain called 33G, genetically identical cells, one has the prion and one doesn't. This is a different genetic strain, one has the prion and one doesn't. And in this case, when the prion is there, the cells acquire the ability, they thrive in the presence of an antibiotic that would normally kill them. In this case, having the prion is not a good thing. And you can see that they're opposite patterns. So it provides a sort of bed hedging strategy for cells to acquire new traits when they're living in complex environments. And we tested about 75 different traits. We tested different carbon sources, different nitrogen sources, all sorts of different things. And basically, this prion, what it does is it gives cells a capacity for growing in different ways. Sometimes that's good, sometimes it's bad. But it allows them to acquire really quite complicated traits all at one go. Because these traits don't generally depend upon the read-through of just one stop codon. They're reading through several different stop codons and accessing several new bits of information. So here's another example of a complicated trait that can be produced, very, very different colony morphology. And this just shows you that every once in a while this happens spontaneously. I told you it's between 1 in 100,000, 1 in a million cells will switch states, and they'll both gain and lose the prion. So allows cells to sample genetic variation on a global genome-wide scale, to acquire complex traits that can depend on a combination of genetic variants in a single step. Now, that was our hypothesis, of course, and you could see that it did give cells a whole bunch of new traits, sometimes valuable, sometimes not. But evolutionary biologists told us, and of course we realized it ourselves, that if this had really evolved for this purpose, if that was the reason why it had been maintained for 800 million years of evolution, it should actually be tuned such that the prion will appear more frequently when cells are under more stress. That is, when they're growing under conditions where things aren't so hunky-dory, and they might need to try out a new trait. They might try that, it might be tuned to try out the trait more frequently. So we measured switching rates in a variety of different conditions, asked whether that happened, and it does. And then you might ask yourself, well, how could that happen? And it's easy, because remember I told you about protein folding. The environment of the cell, the protein folding environment of the cell is so chaotic in there that any kind of stress starts to perturb protein homeostasis, and it makes it more likely that a protein with a prion domain is going to start to misfold in this particular way. And once it gets started, it's going to self-perpetuate. But when the organisms, say, if the cells have the prion state, it's also more likely that they'll lose it. So, because, for example, you induce HSP104 with stress. So it's a very, very sophisticated bed hedging strategy, we believe, in which every once in a while in yeast cells, there's one of the cells will switch into a prion state, it tries out new genetic information. If it's not any good, you lose 1 in a million cells, typically 1 in 100,000 cells, so what? But if the environment changes and that prion happens to give you a new biological trait now that's beneficial, this genotype is going to survive when it otherwise wouldn't have survived. And the whole thing is tuned such that when cells enter stressful environments, instead of switching 1 in a million to 1 in 100,000 times, now they switch 1 in 10,000 times. It's not, they don't all switch. It just makes it more likely that they're going to take a new bet, try something new. And the same thing happens with losing the prion. They lose the prion. It's a little bit more chaotic environment inside the cell, and so they're more likely to lose the prion. And if having the prion is bad, creates a non-adaptive state, they'll try out new ones. So how many of these things are there? Is this just some weird one-off? So we first started testing for that by realizing that the amino acid sequence of that prion determining region was very rich in glutamine and asparagines, and in fact, it's an intrinsically disordered protein region. It doesn't fold particularly in one way or another, and that's what gives us this ability to change its fold and enter into a new prion state. So, but there are lots of, this is sub-35, and Reed Wickner had worked on URI2, which also has a similar kind of domain and can form a prion. We found this one, which also has a prion domain, it's at the other end. So we wanted to figure out, well, there's an awful lot of other proteins in the genome that have these kinds of domains, lots of them. So we tested 96 in the following way. We didn't know what to look for, so we decided we'd borrow sub-30, this other prion protein. We'd substitute that weird domain, all of these other domains, into that weird domain from that protein, and then we should expect, if those proteins can also exist in alternative, heritable states, then we should see those cells, by virtue of these protein, can now switch into the red versus the white state. And by the way, we should also see them forming large amyloid fibers, SDS-resistant fibers. And so of the 96 prion domains we test, prion-like looking domains we tested, we found 25 that actually behaved stably, heritably as new genetic elements. And 24 of the 25 also depended upon HSP104. So HSP104 is controlling the inheritance of all sorts of different traits in yeast. And of course, we had borrowed sub-35 in order to see whether the proteins had this capacity. But what was much more interesting was that when we went back and looked at the endogenous proteins and asked if they could also change their conformational state and create a new form of inheritance, they could. We didn't test every one, but we tested several of them. And many of them created beneficial new traits. Furthermore, what's really interesting is that most of them are RNA binding or DNA binding proteins. So these proteins are positioned in cellular information networks in such a way that they will actually have profound effects on the ways in which genomes are being decoded into new traits. And it provides then an access to lots of new heritable traits. Here's an example of one of the transcription factors that enters into a prion state. And these are the cells growing on rich media. These are the ones that have the prion. They acquire the ability to invade agar. So this is the same colonies as these, except they've been washed off. They've invaded the agar. They also change their ability to flocculate and all sorts of other things. Now, the reaction was mixed when we postulated all of this. And one of the reasons was that, well, wait a second, yeast is the best studied organism on Earth. How come people hadn't found this before? This just doesn't make any sense. Well, I can tell you why, because I took the Cold Spring Harbor yeast genetics course. Everyone who was taking yeast genetics was schooled to do the following thing. When you got a new phenotype, you crossed it to the original cell, and you look for things that segregated in an orderly way, 2 to 2. The things that behaved peculiarly, you threw out. So we really deliberately were not looking for things that it had these unusual kinds of inheritance, and I think that's a really interesting lesson for students. When we're then told, okay, well, wait, if this is true, you should find these things out in the wild strains, and they should be conferring interesting new traits in wild strains of yeast. So we went into the wild. We actually took advantage of other people who'd gone into the wild and had collected yeast strains from all over the world in all sorts of different environments, and we looked to see if they had prions in them. Out of the 700 strains we tested, 31 of them had prions that we could easily look for ourselves, proteins that we had antibodies to. But 250 of them had other protein-based genetic elements which could be cured by HSP104. So prion-like traits abound out in the wild. And by the way, we only looked for 12 traits. So if we found 250 traits, 250 strains that had, just looking at 12 traits, had prion-based, protein-based inheritance in them, my suspicion is that it's a much broader phenomena. So I'm going to finish with a story about another thing that biologists have been ignoring. Although it's really transitioning now, and Jens is one of the guys leading the charge here in terms of microorganisms and the complex biota of our gut. But we always study microbes in isolation. We studied them in isolation, and we studied them growing on pure carbon sources. And they never grow that way in the wild. That's just not what they do. They're always growing in communities. And so bacteria, in fact, are always growing with yeast, and yeast are growing with bacteria. There's just complex communities out there in the wild. And it turns out that bacteria can induce a prion in yeast that is mutually beneficial to both the bacteria and the yeast when they're living together. So let me tell you, and it's a pretty fundamental prion in terms of what it does to cells. So yeast have this amazingly robust phenomena called glucose repression. And it's actually one of the reasons why we love yeast. Because when yeast are growing in the presence of glucose, or even a mixed carbon source, they will only use the glucose. They will turn every bit of the glucose into ethanol for us to drink before they start using up the ethanol. Very fastidious about this. There's a prion that changes that game. It completely bypasses glucose repression, and this peculiar bracket nomenclature basically just means this is a weirdly inherited trait. It's not inherited through normal chromosomal genetics, and we call it GARF, or glucose-associated repression. So all of a sudden, these cells acquire the ability to grow in other carbon sources, using other carbon sources, even when glucose is present. So how do we find it? Well, it turns out someone else had found the phenomena before. They just didn't know what it was. The way they found it was by using a glucose mimetic. So here's glucose, and here's glucosamine. It looks just like glucose, almost like glucose to us. It looks just like glucose to yeast cells. So when yeast cells are grown in another carbon source, they might be growing just fine on that carbon source. But if you put a little bit of glucosamine in the media, they think they're in the presence of glucose, and they won't use that other carbon source. So here's what it looks like. Here are yeast cells growing on glycerol. These are cells, the normal cells that don't have the prion. And if you put a smidgen of glucosamine in the media, they would rather die than use that glucose. The prion, when it's acquired, allows them to grow like gangbusters in the presence of glucosamine. So that's how we selected for this altered state. The cells have their dominant trait. It has cytoplasmic inheritance, all the attributes of prions. It can be cured by changes in chaperone expression, etc. Let me show you an experiment, though, that really makes it interesting. So here's a slab of agar, empty wells. Here are some, every once in a while, spontaneously, the cells will switch into this prion state, and they can grow in the presence of glucosamine using glycerol as a carbon source. If we've taken a strain that we've already selected for that trait, they go like gangbusters. Here's another set. This is just a row of yeast cells that have been serially diluted along this agar plate. Now, what happens when we put a bacteria up here? The bacteria secrete a soluble factor that diffuses across the agar and cause all these cells that were plated here and that were not able to grow before to acquire the ability to grow. But it doesn't, it's a small molecule, but it doesn't get all the way across down here. Is this really what's happening? Well, we can just make conditioned media. We pre-grow bacteria in media. Then we filter out the media. We test for whether or not the filtered media has the capacity to induce this fundamental alteration in metabolism. Sure enough, it increases the rate at which cells switch 100-fold. That was our first fractionation. We now know that when a little bit more pure concentrated, it can cause all of the cells to switch. It's very small. It goes through a 3,000 molecular weight cutoff, filter cutoff. It's extracted into polar solvents. It's stable to boiling, protease digestion, RNase digestion. And my postdoc, who now is going off, Dan Yarosh, who worked on this, is now going off to start his own laboratory at Stanford, knows what it is. So stay tuned. You'll hear about that. It's an interesting story. But GAR persists. So all the cells need is this transient exposure to this environment in which bacteria have been growing. Once they've acquired this new state, it goes on for generation after generation after generation when the cells are cultured in the absence of bacteria. So it's a biological memory state that has been created. It's very, very robust. It's inherited generation after generation after generation. Some of the cells each generation lose it, but most of them maintain it. So here we have a state in which the metabolism of these cells has fundamentally changed. Normally cells use glucose. They churn it into ethanol. They don't get a whole lot of ATP out of it, but they make a lot of ethanol. And it has lots of advantages to have that kind of metabolism because, for example, it'll kill off the bacteria that are in their environment competing with them. But when they switch into this alter, they're capable of getting around this. And this is really, I can't emphasize for how long this has been considered the quintessential metabolic trait of yeast. It allows, this GAR state allows them to use glucose and other carbon sources at the same time. They make much less ethanol, it turns out, but they make lots more ATP. And when you're culturing the bacteria together with yeast, it turns out that the bacteria get a lot of benefit out of it because the yeast are making much less ethanol. The bacterial biomass in co-cultures increases by 100-fold. The yeast get a lot out of it too because they acquire the ability to grow in complex carbon sources when glucose is present. And somehow or other that we don't quite understand, they have much improved longevity when they're growing. When the cells that have the prion are growing together with bacteria, they live much, much longer than the cells that do not have the prion when they're growing with bacteria. So we then started to collaborate with, well, actually, first the thing we did was to look to see whether this was just some peculiarity of our lab yeast, and no, it was present in yeast fungal species that had been diverged for tens of millions of years. It's very conserved. Many, many bacteria have this inducing activity. Question was, does it happen in the real world? And it turns out that it does. In fact, the bacteria that are commonly cultured by vintners who are making wine, that come out of vats of wine that have spoiled, those bacteria turn out to be superinducers. They make a tremendous amount of this compound. And it's a mutually beneficial thing for the yeast cells and the bacteria to have this chemical communication with each other by which they change metabolic states. But it actually acts to our detriment. I'm a little bit hopeful that this may be the way in which I actually get to support my laboratory in the future, is to control this trait. But at any rate, the importance of this for this particular talk is the following. This is chemical communication between a prokaryote and a eukaryote, two different kingdoms of life. It heritably transforms the metabolism of the yeast cells, and it therefore is the ultimate Lamarckian trait because it's basically an environment created by the bacteria, and exposure to that environment creates a new trait that is heritable and robust for generation after generation after generation. So Lamarck was right. And this doesn't mean, of course, that Darwin was wrong. There are logical, reasonable mechanisms by which environment, through its effects on protein folding, can affect the inheritance of new traits. So I just want to finish by just showing you the faces of the bright, wonderful, intelligent, lovely people that I've had the great, great, great privilege to work with and who are responsible for all of that work. So thank you very much.
I think we have time for one or two questions.
So that's my better living through chemistry, by the way. Yes.
Microphone over there. Anybody in charge?
Can prions be transferred through gametes and like sexual reproduction, or can they just be transferred through mitosis?
Yes, the prions can all the ones that we've looked at and that several other people have looked at, Susan Liebman, Yuri Chernoff, Reed Wickner, they are in fact inherited through meiosis. And they're inherited through meiosis in a very peculiar way. When you cross a red and a white cell, the prion state is dominant, and of course it makes sense that it would be dominant because once the template enters into the cytoplasm, is fused with the cytoplasm of another cell, it's going to dominate because it's going to serve as a template for the other cell's protein. But then when the cell goes through meiosis, cytoplasm is passed to all of its progeny. And so all of the progeny wind up inheriting that state. If it was through chromosomal inheritance, you'd see two reds. Looking at the progeny afterwards, you'd see, of meiosis, you'd see two reds and two whites. But you don't. You see the white is spread. And that's actually true for the other prions. So we're now up to, actually, we haven't published on the latest batch, but we're now up to 53 prions. And they all, the way in which we've characterized them has actually been by the fact that they segregate that peculiar way during meiosis.
Thank you.
Peter?
So is this live? Yeah. Susan, that was fabulous.
Could you put this on?
Is this on? Great, thanks. So when you talked about HSP 104, you used the word cut and cleave for disaggregation, but it's not a covalent reaction, it's just a dissociation of the aggregate. So my question is there a lot of biophysics there? That is, if we could understand how aggregated prions get dissociated, when you consider world health problems like Alzheimer, dementia, Huntington's, are there some biophysical principles by which we could understand how to disaggregate things?
Yes.
And could you tell us about that?
Great question. So one of my postdocs, actually, who did a lot of the biochemical work on HSP 104 and showed that it can disaggregate those fibers, it's actually not disassembling them willy-nilly. It looks like it's almost pulling protein out of the middle and winds up severing them. There's still a lot more biochemistry that needs to be done on that. But I'm really glad you asked the question about neurodegenerative disease, because the word amyloid is a word that we all have heard. You've heard about amyloid plaques in Alzheimer's disease, for example. And it turns out that the way this prion protein polymerases is very, very much like the way A beta polymerizes. It first starts to form an oligomeric species, and it's a natively intrinsically disordered protein. It forms oligomers, and they flap around, and they try to figure out. And once they nucleate, once they get the right protein-protein interactions, it goes like this. Now, what's interesting is that after we reported this, it's just another one of these things we had a really hard time getting published because it's a very peculiar way for a protein to polymerize. But once we did, people started showing that these oligomeric species were obligate intermediates in A beta polymerization and in alpha-synuclein polymerization and many other neurodegenerative disease proteins. But what's really important and interesting, and it's actually, I'm so glad you asked the question because it's important in also understanding why these can be beneficial in yeast, is that it's actually not the amyloid fiber per se that's the bad stuff. Those amyloid fibers are pretty inert. I showed you very, very quickly that they can run as SDS-resistant large polymers on gels. They're pretty inert. It's these intermediate oligomers that are the really bad stuff, and that, by the way, is generally becoming more and more accepted. It's not just our idea, it's generally becoming very accepted in the Alzheimer's field. And what's great about the yeast prions is they have evolved to go into that oligomeric state and get right out of it. And if we stop them from doing that, I mean, we've done it in three different ways. We've stopped them, blocked them in the oligomer, and then take that yeast prion and add them onto neurons, they die. So it's the oligomer you don't want.
We break for coffee now, but please come back, and I want to see all speakers here in the panel, and then I'm sure we'll continue